Arguably, cancer is one of modern medicine's largest challenges. Last year alone, over 1.5 million people were diagnosed, and over 500,000 people died from those diseases. Clearly, there's a dire need for new, innovative, and effective treatment methods. I believe marine natural products is an excellent avenue to pursue in this type of research. Actually, about 50% of current cancer drugs on the market come from natural products, so there's already strong support for pursuing this type of research. Luckily in Florida, we're surrounded by the ocean, which is home to a multitude of incredibly diverse species capable of producing secondary metabolites or natural products. Since 1984, Harvard Branch Oceanographic Institute has been building a library of marine natural products they collect from their marine invertebrate and algae samples, which they use to test on a variety of diseases, including cancer. This brings me to my target, Survivin. Survivin is a cancer protein target that is highly expressed in growing tumor cells, but not in surrounding differentiated tissue. This means that if we can find a drug that specifically targets Survivin, we could significantly inhibit the growth of the tumor without detrimental side effects to the rest of the body. Survivin is the smallest member of the inhibitor of apoptosis protein family, at only 16.5 kilodaltons, yet it plays a multitude of roles within the cell, including inhibition of apoptosis and promotion of mitosis. Displayed here is a general workflow of my project. I use marine natural products from my collected samples in an immunofluorescent screening assay. This means I use fluorescent antibody staining specific to my protein target, Survivin, to visualize what's going on within my cells after treatment. I'm able to collect thousands of images for thousands of samples via this semi-automated high-content imager. I then take those images and pull a variety of data that gives me a really complete picture of what's going on within my cells in response to treatment. Once I find a hit, or a compound that significantly reduces the expression of Survivin, I take that compound into the chemistry lab. I isolate, purify, characterize it if it's a novel compound, then take that described compound back to cell biology, where I can use a variety of molecular techniques to determine the exact mechanism of action of how it's able to inhibit the expression of Survivin in cancer cells. I'm confident that these techniques will produce valuable leads in the search for new, effective cancer treatments. Thank you.